In this video, I'll be discussing the development of a quantum sequencing method for applications in precision medicine. This method looks to identify individual nucleotides within single DNA and RNA molecules for sequencing, but also things like structure analysis and also potential applications with other biomolecules like proteins. We hope that this electronic technique can lead to a tool for studying a range of biomolecules on the single cell level, providing information into disease states. A method like this is necessary to combat the highly imprecise nature of modern medicine. So shown here on the left, for the top 10 highest grossing drugs in the US as of 2015, we see that only a very small fraction of people see any physiological benefit from the drug. So for every one person seeing benefit, shown here in blue, there are all the people in red who do not see benefit. So in the best case, we see 1 in 4, but numbers are often as bad as 1 in 20 or even 1 in 25. And the remaining people see no benefit or even negative side effects. That's because these treatments are not designed for addressing the specific biological cause of disease. And this is due to a lack of methods to uncover those specific markers. What we need is a more precision approach that could be shown on the right. So looking at this data, one might wonder why these drugs are selling so much and why we continue to use them. And to answer that question, I'll leave it in the words of a philosopher who stated, "The art of medicine consists in amusing the patient while nature cures the disease." So I'd like to first discuss why a new sequencing method is needed. And this is because current sequencing methods continue to rely on creating millions of copies of a biomolecule sample prior to sequencing, which is time-consuming and expensive, but also leads to a loss of information about the subtle differences between biomolecules within, for instance, a single cell. We need a method that changes the paradigm of these ensemble-based sequencing strategies by eliminating the copying or amplification step, while also having the capability to extract a range of information, such as both the sequence and structure of biomolecules like RNA. So information such as structure would be important due to the role that structure of biomolecules like RNA play in the correct expression of proteins and enzymes inside the cell, disruption of which is a key marker for cancers and other genetic diseases. And so we look to do this by measuring the quantum states of the nucleotides that make up DNA and RNA for their unique identification, and in the future then potentially extend this to amino acids making up proteins. And this can be done with nanoelectronic measurements. So what we hope to achieve is the rapid identification of single biomolecules, where in the final vision we can lay the biomolecule, whether DNA or RNA for instance, down on a surface and interrogate it with electronic measurements that probe the quantum states of the molecule. And then from these measurements, we can extract information on the sequence, structure, and potential biomodifications that are markers for disease. So to uncover the quantum states, we lay down the biomolecule, being DNA or RNA for our demonstrations here, onto a surface and probe the molecule with a tip that creates nanojunctions with the surface. With DNA and RNA, we specifically probe the individual nucleotides, or set of 4 molecules that comprise the overall biomolecule. This is adenine, guanine, cytosine, and thymine in DNA, and uracil, substituted for thymine in RNA. And these are commonly labeled then as A, G, C, and T or U. Now probing comes in the form of a common measurement known as scanning tunneling spectroscopy. So a single measurement consists of changing the voltage or bias V across the nanojunction, of course with the biomolecule in between, while measuring the resulting current I. Now from these measurements we can extract information about the quantum states of A, G, C, and T or U, such as the highest occupied, lowest unoccupied molecular orbitals, HOMO and LUMO. And researchers have performed these measurements on biomolecules in the past with the specific goal, like us, of sequencing in mind. However, when we look at the distribution of HOMO and LUMO states for the 4 nucleotides, A, G, C, and T, shown here, we see that a lot of overlap exists in the values. And so this causes problems when trying to identify nucleotides based on their electronic measurements, which is the ultimate goal of sequencing. So if you take an unknown measurement and see values such as these, for instance, we need more information in order to perform sequencing. And so we sought to remedy this problem by extracting much more information from our electronic spectroscopy measurements, which are inherently much more rich in information than just the HOMO and LUMO that was previously looked at. So we want to form what we call electronic fingerprints, or very unique quantum representations of each of the 4 nucleotides A, G, C, and T or U for DNA and RNA that again contain more information than simply the HOMO and LUMO. So we start by analyzing the electronic spectroscopy measurement in the regions around HOMO and LUMO, where the current begins to take off. And looking more closely at relationships between the current and voltage at both negative and positive biases here, which correspond to hole and electron tunneling, we see significant energetic characteristics. of these measurements, such as what we denote here as the transition voltage, where the tunneling changes regimes, and other features in both low bias and high bias regions. Together then, from these features, we can come up with 6 new parameters in addition to HOMO and LUMO. We next looked at the range just above where the current takes off in our electron spectroscopy measurements. This is characteristic of higher energy molecular orbitals above the LUMO state. So this means orbitals such as LUMO plus 1, 2, 3, etc., which contribute to the overall quantum state of the nucleotide. So to get this information, we look at the spectroscopy measurement above the LUMO level and plot it in a linear relationship. And then from this, the slope and the intercept become 2 additional parameters that characterize a combination of higher energy levels above the LUMO. Lastly, we attempt to quantify the interactions of our nanoelectronic junction with the nucleotides being measured. And we know that the quantum states of the nucleotides can become perturbed and convoluted in a way due to significant proximity interactions with, for instance, the electronic junction tip. So for this, we look in what's called the bandgap region of our electronic spectroscopy measurements. We observe is that the slope in this region, which corresponds to the tunneling conductance, is a good measure of the perturbations that can occur, and so we quantify this. Specifically, when the slope or conductance is large, there are more perturbative interactions between the junction and nucleotide being measured. Vice versa, when the slope or conductance is low, there are fewer perturbative interactions. This tunneling conductance then becomes an additional parameter extracted from our electronic spectroscopy measurement. In total now, including the bandgap, which is the difference between HOMO and LUMO, we have 12 parameters that can be extracted from a single electronic spectroscopy measurement. Now these 10 additional parameters to HOMO and LUMO provide a much more comprehensive fingerprint of the quantum states of the different nucleotides, A, G, C, and T or U. With these new parameters, we are now ready to measure them from single molecules, which we have performed on DNA and RNA. So to collect these measurements, we first lay down a molecular layer on the surface that can help fix or adhere to the biomolecule that's being probed. Now, this fixing is important to restrict unwanted movement of the molecule during measurement, which can perturb the calculation of our quantum states. So with our electronic junction tip, we can visualize the molecular layer and also the deposited biomolecule, right? So we can see that the DNA and RNA are fixed and stretched on the surface with resolution between the individual nucleotides, A, G, C, and T, and U, appearing as separated spheres here in our image. We can then proceed to collect our electronic spectroscopy measurements on top of these nucleotides. So we've all heard of DNA sequencing, but why might RNA sequencing be important? Well, RNA in its sequence is incredibly important as it not only acts as an intermediate between DNA and proteins within the cell, but also leads to catalytic functionalities and processes like regulation and splicing. Currently, there's no direct RNA sequencing methods that are commercially available. Sequencing of RNA relies on first converting the RNA into DNA and then sequencing it, which leads to errors and loss of important genetic information. So a direct method to sequence RNA would be a significant step forward in accurately being able to analyze all cellular processes that involve RNA. Many of these processes of RNA in the cell involve specific structures, and so being able to decode not only the sequence but also the structure would provide additional information that's critical to better understanding the roles of RNA and its importance as a marker for disease. To investigate the possibility of direct RNA sequencing with our method of measuring the nucleotide quantum states, we employ machine learning techniques. We do this as a means of classifying individual electronic measurements as individual nucleotides, A, G, C, and U, otherwise known as base calling. So the machine learning algorithm we use is called a naive Bayes classifier, which takes the 12 parameters that we extract from our electronic measurements and calculates the probability for how closely that measurement resembles each of the 4 letters A, G, C, and U. And it does this by comparing the unknown measurement to hundreds of and thousands of measurements collected on each of the known letters A, G, C, and U. What we have found is that all 12 of the biophysical parameters are often not necessary, and that select sets of them can be used for the best base calling. And for real sequencing applications, and due to the fact that our biomolecules that are surface bound, we can take repeat measurements of the nucleotides to help further increase accuracy. We first analyzed the sequencing capabilities using only HOMO and LUMO parameters, which remember is what previous studies had limited themselves to. So in this scenario, we see accuracies or recalling that is not much better than the case of random guessing, and the situation is not much improved with repeat measurements. Using the new biophysical parameters, which again give more comprehensive quantum state measurements, we see much improved results. So some repeat measurements are performed. We can very quickly achieve identification of AGC and U at levels greater than 93%. Looking at the probability signals which lead to the base calls, we see high resolution, ultimately giving high confidence in the calls, and in turn then, those high accuracies. So this demonstrates the potential of using these nanoelectronic measurements for direct sequencing on single RNA molecules that are not amplified. We now move on to test the potential for RNA structural characterization in addition to sequencing. So methods are available for using chemical labels that attach to RNA in very specific regions of the molecule. We want to be able to detect these region-specific labels and back calculate a structure. A chemical constituent called NMIA is used here to specifically label regions of flexibility in an RNA chain, so such as the stem loop structure as observed with the labels in the schematic. So we need to be able then to detect changes to the quantum states of the nucleotides AGC and U from the addition of these labels. And looking at 2 of the 12 parameters for A, we see that this is the case. There are significant shifts in some of the parameters due to this NMIA label. We can analyze accuracy for the structure analysis in 2 ways now. The first is with combined sequence and structure information. Here we see that the NMIA structural labels can be detected with pretty high accuracy. And similarly, we see high accuracy results for the second case, where we are only interested in looking at the structure. Now, these results demonstrate use of this quantum sequencing technique not only now for sequencing, but probing biomolecules in terms of their structure. I hope that what we've shown here has demonstrated the exciting potential for a nanoelectronic method with applications on single-cell studies in precision medicine. We specifically demonstrated potential with RNA and probing its sequence and structure, which regulates transcription, translation, and a variety of catalytic mechanisms within the cell that are responsible for health, wellness, and also disease states. The possibilities are endless in terms of expanding the surface characterization technique to other biomolecules. So I'd like to thank my fellow group members who were part of this work, specifically Gary, Sepide, and Sajida, and of course also the funding sources who made this research possible.